Welcome to Wildcat Science with Mrs. Haugo. This is Human Biology Genetics, Part 3. And we are talking about genetic diseases, and we're talking about congenital versus hereditary diseases, those two terms. So congenital means present at the time of birth. So clubfoot, which is where the foot kind of turns, the ankle kind of turns in here, that is congenital. It's not hereditary, it just happens in the womb, but it is present at birth. Whereas hereditary means it is genetically transmitted, like you got it from your mom or your dad, it was in your genes. And polydactylism is hereditary. So we can see that this person has 6 toes, and that is hereditary, that is controlled by genes, and it's also congenital. They had 6 toes at birth. So what are some of the reasons? Well, one factor in congenital diseases, those that are present at time of birth, is the lack of an essential nutrient. So for instance, spina bifida is when there is an incomplete closure of the spine, and the spinal cord can actually kind of project through the spine, and that is caused by a lack of folic acid during pregnancy. So that sounds kind of bad. Let's look at a picture where this spinal cord has actually protruded through the spine. And on page 566, there is another picture, which you can see better in your book, and it shows a diagram and then a picture of a real baby, and it actually looks like the baby has like a brain on his or her back because of a lot of the spinal cord development outside of the spine. So that's not good. We want to avoid that. How can we avoid that? Well, since it's caused by a lack of folic acid, by eating things with folic acid in them. Green leafy vegetables, enriched grains, or folic acid or folate supplements. And this is something that happens very early on during pregnancy, perhaps even before a person knows they are pregnant. So if you are a woman of childbearing age, it's a good idea just to maybe take a folic acid supplement. Okay, another cause of congenital diseases besides a lack of essential nutrient is these things called teratogens, which are factors that cause abnormal prenatal development. So one of them is a maternal infection. So if the woman is pregnant during and gets sick during pregnancy, some things can cause abnormal prenatal development. So rubella is a contagious viral infection, and if the mother is sick with rubella while pregnant, often the baby will be born with hearing loss or defects in the brain, heart, or eyes. So that doesn't sound good. One way to prevent that is by having your rubella vaccine, which is a pretty common one to get when you were a baby. So the vaccine is often called MMR, mumps, measles, and rubella, and that's one that people frequently get in childhood, and that's something you can check with your doctor to see if you have. And like I said, it can be prevented by immunizations. So toxins are another type of teratogen or factor that affects normal fetal development. So let's move this up just a little bit. Toxins can damage genes and therefore interfere with fetal development and hurt brain development. So what are some examples of those? Well, radiation, mercury, lead, PCB, which is found in some types of plastics, and etc. A bunch of other things. Drugs. Don't take drugs. especially if you're pregnant. LSD, or acid, can cause chromosome damage. Opioids can cause preterm birth, a low birth weight, and birth defects. So an example of an opioid would be like a fentanyl. Alcohol. So alcohol during pregnancy, we've all seen the warnings like don't drink when you're pregnant. It can cause mental disabilities, physical disabilities, attention and behavioral disorders. So fetal alcohol syndrome is what could happen to if the baby is born with mental and physical disabilities because of the mother drinking alcohol while pregnant. So alcohol is the leading cause of preventable birth defects and developmental disabilities in the United States. So if you are pregnant, don't drink alcohol. Okay, genetic diseases. You are going to teach me some things about genetic disorders. So you're going to pick a genetic disorder and you're going to make like a pamphlet, like something that you would find in a doctor's office. And I want you to start thinking about which one you're interested in doing. Some options are Down syndrome, Huntington's disease, Marfan syndrome, PKU. I don't know if I'm going to practice that one. Let's just call it PKU right now. Sickle cell disease, cystic fibrosis, Tay-Sachs disease, Klinefelter syndrome, Turner syndrome, progressive muscular atrophies, albinism, Fragile X syndrome, osteogenesis imperfecta, neurofibromatosis, cancers linked to certain gene mutations, and if there's one that you're interested in, check with me just to make sure that it fits into our category. So you are going to be telling us about genetic diseases, and while you're busy doing that, let's just jump ahead to the treatment and prevention of genetic diseases. So one way to prevent genetic diseases is genetic counseling. So with this, prospective parents meet with a professional and they look at the family histories of both parents. And they would figure out if the parent is a carrier for certain genetic disorders and what is the likelihood that they will pass that genetic disorder on to their children. And then it gives the prospective parents information so that they can make up their own minds if they want to have biological children or not, or adopt children. Helps them make decisions. Another thing you could do are prenatal tests to screen for some birth defects. The nuchal transparency test, and that is a sonogram. And it looks for excess fluid at the back of the neck, this little nuchal part. And that is characteristic of Down syndrome, and that is something that would happen between 11 and 14 weeks of pregnancy. Another thing people could do to screen for genetic diseases is a maternal blood test. So there's a pregnancy-associated plasma protein test. With the maternal blood test, you're often looking for proteins associated with Down syndrome. Another test you could do is amniocentesis. And with this, you have a small amount of amniotic fluid is removed and then analyzed for chromosomal abnormalities or genetic disorders. And this happens later on, about 16 to 20 weeks. So you actually remove some of the amniotic fluid and some of the baby's DNA would be in there. So then you could look at the chromosomes and see if there are any chromosomal abnormalities. Another thing one can do is chorionic villus sampling. So during that, the membrane of the embryo has these little hair-like projections, the chorionic villus, and you can take some of those projections off, and the baby's DNA would be on there, and so you can analyze that for chromosomal abnormalities or genetic disorders. And the benefit of this is that you can do that much earlier, 8 to 10 weeks, compared to 16 to 20 weeks. There is a karyotype. So any of the cells obtained by the chorionic villus sampling or the amniocentesis are grown, and they are stopped at metaphase of mitosis, and they're photographed, and then that's when you can most easily see the different chromosomes, and so then you could look for chromosomal abnormalities then. Okay, we're going to learn more about Benn's cystic fibrosis. And you don't need to write this down, but we're going to be learning more about gene expression and the lac operon, kind of more about gene expression and some molecular biology and how your genes are regulated. And I'm hoping to go through, we'll have time for our forensics lab. And if we have time, we'll do some epigenetics. So your genetic notes are done. Good job.